Dear students, today we are going to start another important topic of biochemistry, and that is entitled as Biochemistry of Digestive Tract. Under this topic, we are going to see these contents in detail. The very first one is introduction, chemical composition, secretion, and regulation of different types of digestive juices of GIT, including saliva, gastric juice, pancreatic juice, bile, and succus entericus, which is also called as intestinal juice. Then we are going to see that how carbohydrates, lipids, proteins, and nucleic acids get digested or hydrolyzed in gastrointestinal tract. Afterwards, we would see in detail the mechanism of absorption of carbohydrates, lipids, and amino acids in the digestive tract. The second last portion of this topic would be focused on disorders or diseases associated with GIT. For instance, achlorhydria, peptic ulcer, lactose intolerance, cholelithiasis, pernicious anemia, cystic fibrosis, and celiac disease. And the very last portion, it would be focused on studying site of synthesis and major actions associated with different gastrointestinal hormones like gastrin, cholecystokinin, secretin, gastric inhibitory peptide, vasoactive intestinal polypeptide, mutilin, and enkephalins, substance P, neurotensin, and enteroglucagon. Well, this is the snippet of table of specifications where you can see the weightage of SEQs and MCQs for this topic. Now you can spot here this topic at number 9, which is biochemistry of digestive juices of GIT, digestion and absorption in GIT. Now the weightage of SEQs for this topic is 0.5, which means 2.5 marks. And then the weightage of MCQs for this topic is 3, which means that 3 MCQs each carrying 1 mark. In today's lecture, we are going to see introduction, chemical composition, secretion and regulation of saliva, gastric juice, and pancreatic juice. The very first digestive juice of GIT that we are going to see is saliva. The word saliva has been derived or originated from Latin language that means to spit, spittle, dribble, drool, etc. It's basically an aqueous medium or mixture that contains mucus and enzymes. By definition, saliva is basically a watery secretion or suspension which is produced in the oral cavity that helps digest food. Now, what are the characteristic features of saliva? It appears thick, colorless, opalescent, or milky, or slightly turbid, and slimy. Its pH, when it is freshly secreted, ranges from 6.4 to 7.1, but during the process of active secretion, it may reach up to 7.3. Now, what is its volume capacity? In an hour, saliva can be produced up to 20 ml, and in 24 hours, the volume of saliva that is produced ranges from 1000 to 1500 ml. That means 1 to 1.5 liters. Its specific gravity is 1.002 to 1.008, which shows that this is highly hypotonic if it is compared with blood plasma. Saliva is mainly composed of water, and that is around 99.4%. Rest of saliva contains solids which constitute about 0.6% of total solids, and these are further distributed as inorganic solids which are up to 0.2% and organic solids which are up to 0.4%. Now the composition of saliva, it is highly variable, and this varies according to the stimulus it receives, which can result into the secretion of saliva. During ingestion of meals, rate of salivary secretion increases, while during dehydration, salivary secretion decreases, which can further increase the sensation of thirst. In saliva, inorganic solids that are found mainly include sodium, potassium, calcium, magnesium, chloride, and bicarbonate. But in minor amounts, some other inorganic solids can also be found in saliva, like sulfate, nitrate, iodide, and thiocyanate. Especially this thiocyanate is found in the saliva of smokers. As far as organic solids of saliva are concerned, so in saliva, amylase is the key enzyme which is found, and this is the main organic solid of saliva, and this is helpful for the digestion of starch. Another important organic solid that is found in saliva is mucoprotein, for example mucin. And the other components that may be present as organic solids in saliva include lactic acid, ascorbic acid, choline, phenols, urea, glucose, etc. Other organic solids that can be found in saliva include cholesterol. Then traces of some enzymes can also be seen in saliva. For instance, phosphatases, then lipid digesting enzymes like lipases, carbonic anhydrase, then an antibacterial enzyme called muramidase. This is also known as lysozyme. Kallikrein, this is a protein which is hypotensive protease, and this is used to regulate blood pressure. Then a defense protein called immunoglobulin A. So all of these are found in traces in saliva. Salivary enzymes act on starch and hydrolyze starch, but salivary enzymes are not capable enough to break down starch completely. Complete digestion of starch takes place in the small intestine. In stomach, action of salivary amylase comes to an end, and this is due to acidic environment of the stomach. Secretion of saliva is done by three important salivary glands. For example, parotid gland, which is a type of serous gland, and this is rich in alpha amylase. Then the second one is submaxillary gland or submandibular gland, and this is rich in mucus, and the chief protein which is found in mucus is called mucin. And the third is sublingual gland, which is also rich in mucus. Now, the amount of secretion and composition from each salivary gland, this is variable, and this is basically dependent on two important factors. One is the type of stimulus of the food, and then the nature of the food. Actions and functions associated with saliva. The very first function of saliva is this, that it provides lubrication. It helps chew and swallow food, and this is also helpful for making the food moistened. It assists in the preparation of food for its further digestive action in gastrointestinal tract. The other main function of saliva is its digestive action on starch with the help of its enzyme, which is called alpha amylase, also called as ptyalin. This enzyme acts on starch and converts it into dextrin. Then in the small intestine, it is further digested to maltose, and eventually it is converted to glucose. There are three types of amylases that we can find in human beings, and that is salivary amylase, pancreatic amylase, and intestinal amylase. Salivary amylase can only work on cooked starch, and pancreatic and intestinal amylases can work on both cooked starch and uncooked starch. There is another enzyme which is found in saliva, and that is called lingual lipase. And this enzyme is responsible for hydrolyzing short-chain dietary fats. The third most important function associated with saliva is its cleansing function or its cleaning function. It basically clears buccal cavity from microorganisms, and as a result of which it maintains overall hygienic condition. There is one specific enzyme which is found in saliva, and that is called lysozyme or muramidase, which is having antibacterial activity, and therefore it helps kill bacteria. And as a result of its cleansing function, it basically prevents the development of dental caries. Solvent function of saliva. Saliva helps dissolve food material, It activates taste buds to stimulate appetite, and thus it promotes further secretion of saliva. It can also activate the secretion of gastric juice in the stomach. Then its moistening action may also assist in articulation of speech. Excretory function associated with saliva. Many substances or compounds, both inorganic or organic in nature, may be excreted through saliva from the body, and these may include metals, for example lead, mercury, then drugs, alkaloids, for example morphine, antibiotics, for example penicillin, and even different or several types of viruses that may cause rabies, mumps, polio, AIDS, etc. These all substances can take exit from the body through saliva. Saliva also maintains water balance, as we have seen earlier that during dehydration phase, saliva gets dried out or it gets desiccated, and as a result of which it can increase sensation of thirst. It is also helpful for deiodination, and as a result of which it regulates thyroid hormone. It is also involved in esophageal clearing, and thus it clears esophagus from any refluxed material that is coming from stomach. Regulation of saliva takes place by three important mechanisms. First is via unconditioned reflex, then conditioned reflex, and the third one is spontaneous reflex. Unconditioned reflex, as you know, that it takes place unconditionally, naturally, and automatically. So basically it occurs when any material is placed in the mouth. Even in an empty mouth, movement of tongue can also trigger or stimulate this kind of salivary secretion. Then the second type of reflex that can also regulate the secretion of saliva is conditioned reflex. Major stimuli which are responsible for the onset of this kind of reflex are sight and smell. Efferent impulses that arise from esophagus and stomach may generate this type of salivary secretion. You can relate here the experiment that was conducted by Russian scientist Pavlov in dogs. He basically studied the stimulation of saliva in dog's mouth when he sounded bell while presenting food to dogs. Then the third type of reflex that can also control the salivary secretion is spontaneous reflex, and this occurs without any interruption, or it takes place continuously even when there is no stimulus, and it basically keeps pharynx and mouth moistened, and this is believed to be stimulated or triggered by neurotransmitter acetylcholine. Okay students, we are going to start another important digestive juice which is called gastric juice, and this is basically a mixture of different types of digestive secretions that are produced from the stomach. Now what are the characteristics or properties of gastric juice? So it basically appears colorless whenever it is uncontaminated, but if it is contaminated then its color may vary. Its pH ranges from 0.9 to 1.5, and its volume capacity is 2000 to 3000 ml or 2 to 3 liters per day. Now its chemical composition, it is mainly composed of water, and that is around 99.4%, while the inorganic or organic matter that is found in gastric juice is around 0.6%. Inorganic matter that is found in gastric juice is mainly composed of HCl, which activates pepsinogen to pepsin. It lowers pH, therefore it favors action of pepsin, that is hydrolysis of proteins. It has also got antiseptic function, and thus it clears or kills microbes. It helps absorbing iron, and it also increases the levels of secretin protein. Other inorganic ions which are found in gastric juice include sodium and potassium, and the ions which are found in traces in gastric juice are calcium, magnesium, phosphate, and sulfate. Organic matter of gastric juice is composed of mucin, which is a type of glycoprotein. It assists in lubrication process. It basically forms protective layer over mucosa, and thus it shields mucosa from the damaging effects of gastric juice. It has also got anti-pepsin activity, and it also neutralizes HCl. Protein digesting enzyme which is found in gastric juice is pepsin. Lipid digesting enzyme gastric lipase is also found in gastric juice. Then a glycoprotein called intrinsic factor is produced from parietal cells of stomach, which helps in the absorption of vitamin B12 in the ileum of small intestine. Lactic acid in traces can also be found in this digestive juice, and if it is found in excess, then it indicates bacterial growth or bacterial infection. Then another protein called renin is found in gastric juice of infants or cattle. This is also called chymosin or milk curdling enzyme. Then gelatin hydrolyzing protein or enzyme gelatinase, gastric mucus, lysozyme, which is also called muramidase, a type of antibacterial enzyme, carbonic anhydrase, and urease are also found in gastric juice. Here is the list of some important enzymes that are found in gastric juice, which makes up the important part of organic composition of gastric juice. So these enzymes are pepsin, gelatinase, renin, and these three are all types of proteases, and then lipid digesting enzyme called gastric lipase. Now, secretion of gastric juice is done mainly by three types of cells which are present in the gastric glands, and these are called chief cells or zymogen cells, then oxyntic or parietal cells, and the third category is mucus cells. So this table is showing the cells and then their secretory products and their associated functions in different areas of stomach. For example, this part is the first part of stomach or the upper portion of stomach, which is called as cardiac part of stomach. And the cells which are found in this part of stomach are mucus cells, and their secretory products are mucus and bicarbonate, and their main function is protection and lubrication. Then the middle part of the stomach is called as its fundus and body of stomach, which contains these types of cells. Now from the parietal cells, which are also called oxyntic cells, the secretory products are HCl and intrinsic factor, and this HCl, it basically helps denature proteins and it activates pepsinogen, while intrinsic factor is involved in the absorption of vitamin B12. Then the chief cells, which are also called peptic or zymogen cells, these are involved in the secretion of pepsinogen, which aids in protein digestion process. And the mucus cells produce definitely mucus and bicarbonate and thus provides lubrication and protection. Enterochromaffin cells secrete serotonin, which induces peristaltic activity. These cells in the fundus of stomach produce somatostatin, and this basically decreases the secretion of HCl. Now the lower portion or the last part of stomach, which is called pyloric or the antrum, that basically contains mucus cells, which produce mucus, and bicarbonate, thus providing protection and lubrication. And then this part also contains G cells, which are involved in the secretion of gastrin, thus promoting HCl secretion. D cells in the pyloric part of stomach can produce somatostatin, and thus they can decrease the secretion of HCl. Then enterochromaffin-like cells, which are abbreviated as ECL cells, produce histamine, which also triggers HCl secretion. Secretion of gastric juice or HCl, it can be maintained even when stomach is empty. In post-meal consumption, its secretion increases. In response to meat, again its concentration or secretion becomes increased. While in response to bread or milk, there is less secretion of gastric juice. Now we are going to see stimulants that can trigger or induce gastric secretions. Examples of stimulants include alcohol, alkalis when consumed in small amounts, caffeine, then hormone cholecystokinin, which is also called pancreazymin, corticosteroids, for example cortisol, gastrin, gastric irritants, for example spices or some condiments, parasympathetic agents, for instance acetylcholine, histamine, nicotine, and even some physiological factors, for example stress, anxiety, fear, etc., can all trigger gastric secretion. Now we are going to see examples of depressants or inhibitors of gastric secretion. The very first example is fats. Fats may suppress gastric secretion because fats are mainly digested in intestine and there are no enzymes except gastric lipase in stomach, and gastric lipase can only work on short and medium chain fatty acids. Then another depressant of gastric secretion is acid. For example, when acidic food it touches upper duodenum, then there is release of secretin hormone that can increase the level of bicarbonate while it decreases HCl secretion. Then the other examples include alkalis when consumed in large doses. Then parasympatheticolytic agents, for example some drugs like atropine. This drug can also inhibit gastric secretion as it can inhibit or block the action of acetylcholine. Then surgical procedures, for example vagotomy, in which vagus nerve is truncated and thus HCl secretion can be inhibited. Then some anti-gastrin drugs like proglumide and some proton secretion blocking drugs, for example proton pump inhibitors like omeprazole. Then antihistamine drugs, for example ranitidine, which is also sold under the name Zantac. Then secretin, prostaglandins, somatostatin, and gastric inhibitory peptide, which is released in response to fats and exoses in intestinal mucosa. These all can depress gastric secretions. One of the important aspects associated with prostaglandins is this, that there are some protective prostaglandins which inhibit HCl production and thus increase the concentration of bicarbonate. But whenever NSAIDs are taken excessively, for example non-steroidal anti-inflammatory drugs like ibuprofen, brufen, etc., then these NSAIDs intake may result into lowering of prostaglandins, and as a result of which HCl secretion increases and it may increase the risk of ulcer. Here is the mechanism of HCl secretion given, and you can see the production of HCl from parietal cells. So basically what happens that carbon dioxide react with water in the presence of carbonic anhydrase and you get carbonic acid. This carbonic acid undergoes spontaneous dissociation into free protons and bicarbonate. These protons are released into gastric lumen in exchange with potassium ions with the help of proton pump, which is also called as proton potassium ATPase, and here one ATP is utilized. Now these bicarbonate ions are released into blood, and these are exchanged with chloride ions, and chloride ions are now available in parietal cells. Now this exchange takes place with the help of anion exchanger, and these chloride ions are further sent into gastric lumen with the help of chloride channels, where chloride ions combine with proton to make HCl. There are three phases or mechanisms by which regulation of gastric juice secretion can be done. One is neural or cephalic phase, which is stimulated by smell, sight, and taste of food. Then we have gastric phase, which is triggered or stimulated when the food is present in the stomach. And then the intestinal phase is triggered whenever there is direct introduction of food into the duodenum. For example, if the chyme presented to duodenum is fatty and definitely its pH is low, then it can result into the release of secretin hormone, which increases the level of bicarbonate while it decreases gastric secretions. Now we see important types of receptors that regulate gastric juice secretion. For example, acetylcholine, histamine, and gastrin receptors stimulate the production of HCl, while prostaglandin and somatostatin receptors suppress HCl secretion. Now the third digestive juice we are going to study is pancreatic juice. This is basically a mixture or suspension of different types of digestive secretions which are produced in response to acidic chyme in the upper part of duodenum. Now what are its properties? Pancreatic juice appears as clear watery liquid or secretion, and its pH ranges from 7 to 8.3, and per day human beings produce 1.2 to 1.4 liters, that is 1200 to 1400 ml, and its specific gravity is isoosmotic with blood plasma, and that is in between 1.010 to 1.018. Chemical composition of pancreatic juice. Pancreatic juice is mainly composed of water that comprises around 98%. Then the rest of the pancreatic juice is composed of solid portion, and that is around 1.8 to 2%, which is further distributed as organic matter and inorganic matter. Inorganic matter of pancreatic juice mainly constitutes bicarbonate ions, chloride ions, sodium, potassium, while in traces you can find phosphate, calcium, and magnesium. Now, organic matter of pancreatic juice is mainly composed of amylase enzyme, which is also called as pancreatic amylase, and then trypsin and trypsin inhibitor, chymotrypsin, carboxy peptidase types A and B, then elastase, collagenase, lipase and colipase, then phospholipase A2, then esterases, for example, cholesterol esterase, DNAs, RNAs, and some others that can be found in organic matter of pancreatic juice are immunoglobulins, then kallikrein, lysosomal enzymes like muramidase, then alkaline phosphatase, albumin, and even lecithin. Functions of pancreatic juice. Since pancreatic juice is alkaline in nature, so therefore it can neutralize or buffer acidic chyme that arises from stomach. It constitutes mainly digestive enzymes, and it has enzymes for digesting or hydrolyzing carbohydrates, lipids, proteins, and even nucleic acids. Now the very first enzyme that we are going to study, which is found in pancreatic juice, is amylase. Amylase or pancreatic amylase is able to convert dietary starch and glycogen into these partially hydrolyzed products. For example, maltose, maltotriose, and dextrins. And remember that amylase is activated by chloride ions. Then trypsin and chymotrypsin are proteolytic enzymes which are found in pancreatic juice, and these are also termed as endopeptidases, because these enzymes can cut proteins or peptide bonds from within the protein structure. These trypsin and chymotrypsin proteolytic enzymes are synthesized in the form of their inactive precursors, which are called zymogens. For example, trypsin is found in trypsinogen form, and chymotrypsin is found in chymotrypsinogen form. So trypsinogen, which is the inactive precursor of trypsin, is converted into its active form trypsin with the help of enterokinase enzyme. And then this trypsin becomes able to hydrolyze or break down peptide bonds which are formed or established by basic amino acids. Chymotrypsinogen is an inactive form of chymotrypsin, so it is converted into chymotrypsin with the help of trypsin, and it can hydrolyze peptide bonds which are formed by uncharged amino acids. Then carboxy peptidase is another proteolytic enzyme which is found in pancreatic juice, and this is exopeptidase, which means that it cuts proteins from the terminals or from the termini of proteins. It is synthesized as its inactive precursor, which is called procarboxy peptidase. This procarboxy peptidase in the presence of trypsin is converted into carboxy peptidase, and it becomes active, and now it can hydrolyze peptide bonds which are present at the C terminus of protein. C terminus is that contains carboxylic acid group of amino acid. Then another proteolytic enzyme that we can see in pancreatic juice is elastase, which is synthesized as proelastase, but in the presence of trypsin, it gets activated to elastase. And it has the ability to break down elastin and some other related proteins. Then collagenase enzyme that can split collagen, it is also found in pancreatic juice. Pancreatic juice also contains lipase that can hydrolyze fats, and it needs colipase. With the action of pancreatic lipase, fats or triacylglycerols are converted into diglycerides, monoglycerides, fatty acids, and glycerol. Then another lipid digesting enzyme is phospholipase A2, which is found primarily as prophospholipase A2. Now it can hydrolyze phospholipids. For example, it can convert lecithin into lysolecithin, and it can also hydrolyze cephalin into lysocephalin. Now cholesterol esterase, which is the enzyme to hydrolyze cholesterol esters into free fatty acids and cholesterol, is also found in pancreatic juice. This digestive juice also contains some non-specific esterases that can work or act on fat-soluble vitamins and some phospholipids and can hydrolyze them. Even DNases and RNases are also found in pancreatic juice that can hydrolyze DNA and RNA into their respective nucleotides. Secretion of pancreatic juice. Acidic chyme, whenever reaches duodenum, then it results into the release of secretin and cholecystokinin hormones from duodenum. Under the influence of cholecystokinin, there is release or secretion of digestive enzymes from the acinar cells of pancreas. Under the influence of secretin, bicarbonate ions are released in large quantity from the small ducts that arise from acini. These bicarbonate ions can neutralize or buffer acidic chyme and thus can prevent the onset of duodenal ulcers. Remember that stimulation of vagus nerve can also induce enzyme-rich pancreatic secretions. Regulation of pancreatic juice. First, we are going to see its nervous regulation. During cephalic and gastric phases of stomach secretion, there is transmission of parasympathetic impulses that takes place along the vagus nerves that ultimately liberate acetylcholine. And followed this process, there is secretion of large amount of enzymes into acinar cells of pancreas. Hormonal regulation of pancreatic juice secretion. As we have seen earlier, that whenever acidic chyme it reaches duodenum, then it can result into the stimulation of secretion of two important hormones, that is secretin and cholecystokinin. Secretin basically stimulates the production of bicarbonate ions, while cholecystokinin is involved in the release of digestive enzymes. Okay students, activity for you. You are supposed to recall today's lecture and give the answers of these two queries. I'm going to reveal the answers.